So I am your MC for this evening. You'll be getting that $10 later then. Robin, you missed out on 20. If you'd get him bigger shite, I'm just saying. My name is Alan Duffy. I'm an associate professor of astrophysics at Swinburne University. I am a scientist, first and foremost, but I'm a scientist who also believes passionately in communicating that science. And that's why I couldn't be prouder than I am to be the MC of this incredible event. FameLab, as we'll hear, is truly unique, and in terms of its scale, absolutely is the premier science communication competition in the world. You'll hear more from me in a little bit, but to get us started this evening, I'd like to welcome to the stage the British High Commissioner to Australia, Her Excellency, Menna Rawlings, CBE. Please make her feel very welcome.
Thank you very much, and good evening, everybody. I'd like to thank Mr. Maguire for his generous welcome to country, and I pay my own respects to the traditional owners of this land, elders past and present, and extend that respect to other Aboriginal and Torres Strait Islander people here with us tonight. It's a pleasure to open this year's final of FameLab, the world's leading science communication competition. I was last here 2 years ago, and it's great to be back. I'd like to share with you a quote from the late, great Stephen Hawking, one of the most brilliant scientific minds we've ever known, and who did perhaps more than anyone to communicate the power of science to the public. Professor Hawking once said, "For millions of years, mankind lived just like the animals. Then something happened which unleashed the power of our imagination. We learned to talk, and we learned to listen. Speech has allowed the communication of ideas, enabling human beings to work together to build the impossible. Mankind's greatest achievements have come about by talking." So the communication of ideas and the power of imagination are exactly what tonight is about. I can't wait to hear from our amazing lineup of finalists, and I promise not to talk too much myself, bearing in mind the criteria against which tonight's presentations will be judged: content, clarity, and charisma. Scientific collaboration is one of the most exciting aspects of the modern dynamic partnership between the UK and Australia. Our universities and research agencies produce more than 6,000 co-publications annually, working together in fields as diverse as Earth observation, sports science, meteorology, and defence technology. And I'm very proud of the British High Commission's role in support of FameLab, working with the wonderful British Council, Helen Salmon and team, who do such a great job putting it on across this land. And the partnerships they have developed in Western Australia enable us to make this very special event happen. And it's a great example of how government, by working with civil society organisations, businesses and universities, and cultural institutions, can really do great things together. So a brief word about FameLab. I'm sure you know all this, but just a quick recap. Founded in Cheltenham in 2005, FameLab has seen 9,000 young scientists and engineers participate in over 30 different countries. In Australia, this competition is just one part of the British Council's work to create people-to-people links between international leaders of the future in science, in education, and the arts. And although we call tonight a final, it's really only the beginning for this group of up-and-coming researchers. Our finalists have spent 2 intensive days undergoing training with the legendary British science communicator, Malcolm Love. This week, they will present to school children here in Perth, and from there, they will embark on a journey of science advocacy, becoming ambassadors for Australian science and innovation. We've been thrilled to watch Dr. Neural Coxeton, the winner of last year's National FameLab final here in Perth, make it all the way to Cheltenham to be global runner-up. I met Neural earlier this year and heard about her amazing research into the antimicrobial. I'll try that again. Maybe I won't, actually. I'll just carry on. Properties of honey. I haven't looked at my morning crumpet the same way since. Neural's growing career and profile are an inspiration to us all. Many of you in the audience will be thinking about future careers, and you may have heard some scaremongering reports about future jobs being lost to robots. Today's technology is simply amazing. Perhaps one day you'll see a robot high commissioner. But I'd like to offer some reassurance. A recent report from the British Innovation Foundation, Nesta, showed that the opportunities of the future will be for people that combine creative and STEM skills. These are the things that robots can't do, and that will enable us to do what Stephen Hawking challenged us to do, to work together to build the impossible. Ladies and gentlemen, please enjoy tonight's presentations and be inspired by the power of imagination. Thank you.
Thanks so much, Your Excellency. And now it may, well, I hope it's not escaped your attention that we're sitting at the University of Western Australia, and we are very proud and delighted to be here and hosted by this university that was actually the first place I arrived at when I came a decade ago as a starry-eyed, immigrant to this great country. And it was at this university that I had the most traumatic experience of my life when I saw a black swan with red eyes. It was harrowing. I don't know where I'm going with that, by the way, Professor. So to say some words as Vice-Chancellor of the University of Western Australia, could you please welcome to the stage Professor Dawn Freshwater.
Good evening, ladies and gentlemen, and can I start, as I always do, by also acknowledging that we meet on the traditional owner's land, that is the Whadjuk people of the proud Noongar Nation. I'd also like to acknowledge that we do have peacocks on this campus, as well as swans that are around by the river, and those peacocks are actually not as scary as I'm sure the swans were to Professor Duffy when he first arrived. It's a great pleasure to be here this evening, and particularly to see so many people in the audience, first of all, because normally when I stand on this stage, it's with an all-staff meeting and I'm talking about strategy, and it's quite hard to get a full audience. But secondly, because I can see so many of our friends, colleagues and distinguished guests here this evening, and it's great to welcome you also. So I'll just mention one or two of those, and thank you to the High Commissioner, Mena, for your opening there and to remind us about the importance of communication. Science communication actually is one of the things that's really high on the agenda for universities at the moment. I'd also like to welcome the Honourable Tony Butty, the MLA member for Armidale. Of course, we've heard from our MC, Alan, Lynn, great to see you on campus as always. Robin Williams, who I met for the first time this evening and was pondering the issue of diversity in Vice-Chancellors in Western Australia and how we've managed to achieve it. So, great question. Helen Salmon, the Director of the British Council in Australia, I'm very pleased to have you here. I've done a lot of work with the British Council myself. Great to be doing something co-sponsored. And of course, importantly, our FameLab national finalists. And may I just say, as the Vice-Chancellor of this university, it's a privilege to be involved in this, what is really the largest science communication competition in the world. FameLab provides us with an opportunity to come together to learn about the inspiring research that our finalists are working on in a creative and a compelling way, which we need that narrative at the moment in society to help build that bridge. So much of what we do in terms of the communication of many areas of activity across the university is broadcast, of course, internationally and nationally. But this personal experience that we can have in this auditorium tonight of a compelling narrative is really one that is quite meaningful. And I think we all need those to remind us about what we're doing, why we're doing it, and how we can improve lives in this way. So firstly, for me to congratulate all the contenders for making it just to get here this evening to the final. You're competing with the best and the best upcoming researchers from around the country, and just to get here is an incredible achievement, so well done. I'd also like to acknowledge our esteemed panel of judges. We all have enjoyed long and varied careers ourselves, but the judges have experienced particularly careers in science and communication where they bring together collective expertise to the panel to select the best live science communicator for 2018, and that person will go on to compete against the rest of the world. It is, as you know, I'm sure, an imperative that we continue to make science more easily accessible to the public. And one of the things that I've been concerned about for some time is that translation and impact. And certainly with my own PhD students and through the research that I've been engaged in, it's been important for me to have the people that I work with able to talk about their research at a bus stop or at a train station to any member of the public who asks a question in a way that's really accessible and helps them to really understand what it is that's at the essence of what they're doing. Showcasing our research makes a meaningful difference to everyday lives, whether it's the latest medical research breakthrough, which will allow us to live longer and healthier, or sustainability in the work of agriculture, which is helping to feed the world. While our scientists may carry out some of the world's most important research, they need to be able to explain it. As we heard from the British High Commissioner, important that we can continue to carry that communication skill forward as we evolve. Otherwise, how will society know that what we're doing is important? In the words of Einstein, you don't really understand something unless you can explain it to your grandmother. Testament to the University of Western Australia's culture of innovation, I'm proud to say that we have 2 UWA finalists competing in this year's final. UWA's James Wong and Ben McAllister from the ARC Centre of Excellence for Engineered Quantum Systems will speak about vastly different subjects from dark matter to animal biology. Yet their research provides equally exciting opportunities for discovery and progress in science, technology, engineering and maths. I'm certain we'll find theirs and all the presentations compelling. So finally, good luck to all the finalists. I look forward to hearing more, as we all do, about your incredibly varied research projects, spanning from the collection of whale snot, yes, I'm sure you're going to be very interested in that, using drones to the study of how lungs and airways work in hopping kangaroos. Enjoy the evening, everyone.
Thank you very much, Vice-Chancellor. Our final VIP who will say some opening words is the member for Armidale, MLA, Dr. Tony Butty. Would you please make him feel very welcome to the stage?
Thank you very much, Alan. I'd like to commence by acknowledging the traditional owners of the land we stand on and respect their elders past and present and their traditions. It's actually a privilege to be introduced by Alan. Could I watch the Channel 24 most mornings, and Alan's often on there, so I find out a lot about my science through his communications. And I believe he's just become a father quite recently, so I think a round of applause is in due for Alan. I'm glad that, Dawn, you acknowledged everyone that I was going to acknowledge, so that will save me having to acknowledge everyone, but I would like to add a couple people, and that's obviously to the Honourable Malcolm McCusker and Tonya McCusker. Welcome very much for being here. Also to Alec Coles, the Chief Executive Officer of the West Australian Museum, and Mr Maguire for the beautiful welcome to country. I'm representing the Minister for Arts and Culture, or Culture and Arts, David Templeman. He put in his apologies, but today we actually handed down the state budget, so unfortunately he has other commitments tonight. There is a prepared speech here, but I don't want to be a robotic politician, so I'm not going to read it. And I just want to relay a story why we are here in Western Australia to witness this fantastic competition. And of course, the reason that we are doing that, we have in our midst, of course, one of the great scientists in West Australia, and that's Professor Lynn Beasley, who actually, I believe the story goes that she was speaking to the Honourable Malcolm McCusker, I think when he was Governor of Western Australia, if my memory's right, and Lynn had just come back from the UK, had been to the Cheltenham Festival. and was talking about this fantastic competition, and I think was lamenting that she needed to obtain some funding and probably was going to be walking up and down St George's Terrace. And if you're going to law firms, you weren't going to have much success, I wouldn't have thought. As being a former lawyer, I know these things, that they don't like putting their hands in the pocket. But of course, Malcolm, being the generous person that he is, offered to help with the funding of this great competition. So really, the reason that it's come to WA is because of Lynn and also Malcolm McCusker. So thank you very much. And I should also add that the sponsorship of Woodside is also very, very much acknowledged and welcomed by the government, and also the WA Museum is a proud partner in this competition. And don't want to be political, but the West Australian government and the minister is a very strong supporter of this competition. And if the funding needs to be renewed, I hope that you put pressure on the minister. I'm not being the minister, so I can say that. One other thing before I leave the stage is that you will all give yourself a round of applause, because as far as I'm aware, that this is the largest live audience ever to watch FameLab in the world. So put your hands together. In a previous life, I was a science teacher. I look forward to the great communications that we're going to hear tonight of the great ideas by these young scientists. So good luck to everyone and have a great night. Thank you.
Thank you very much, Dr. Putty. So, we begin, but not before some more housekeeping. First off, you're all here because FameLab is incredible. It is the largest science communication competition in the world. It's run by the British Council, and it has operated since 2005, consistently growing throughout that time. For some nations that take part in this competition is truly extraordinary. Egypt has something like 1,400 scientists who've applied to be part of this event. I was just absolutely blown away that over 500,000 views of these talks have taken place in this last decade or so. This is a world-renowned competition, and we should be very proud and privileged to have it here in Perth, and I would like to second the thanks to all of the supporters who've made that possible. Yeah, go on, give me a clap. I will say their names in full, though, so we'll have to continue a round of applause for that in a little bit. Firstly, though, what is FameLab? FameLab is, well, the art of telling your science with anything but jargon. And there's no PowerPoints, there's none of the props, the tools that traditional scientists use when telling their science to other scientists. Instead, there's props and tools that science communicators use, and there's the difference. We're trying to communicate the science as best we can, not necessarily dump a heap of facts onto you. These are creative and super fun ways to tell that scientific story, and I am going to take a seat right there so I get to watch them all because they are so much fun. Now, over the past 2 months, the British Council has traveled the nation, hosting semi-finals across the states. We have whittled down in a brutal series of to-the-death-ish fights to our 11 finalists this evening. To decide between them is truly a brutal and horrific task, and one that is not mine, and that is fantastic. It is, however, these three lovely hanging judges. We have science journalist and broadcaster Robin Williams, presenter of the RN's The Science Show, Occam's Razor. Give a round of applause if you've ever heard of those. And my favorite fact, literally a national living treasure, voted in 1987. I'm serious, look this up. It is actually a term, a national living treasure, phenomenal. All right, then we have Professor Lynn Beasley, West Australian of the Year, former Chief Scientist of Western Australia, a neuroscientist, and also one of the best buddies I've ever had to be beside me to confront masses of school kids and try to convince everyone from literally 3 years old all the way up to teenagers who are too cool for school that science really is awesome and inspirational. Please make Lynn Beasley feel very welcome. And finally, Director of the British Council here in Australia, boss of all of this that you see, and Fellow of the Royal Society for the Encouragement of Arts, Manufactures and Commerce, Helen Salmon. Please make her feel very welcome. So the idea of tonight is that we have identified scientists who are great communicators. They're passionate, willing, they want to tell their science. But they don't want to stop here. It's not enough to just tell all of Australia, they want to tell the world. And that's exactly what's going to happen to our winners who will compete in the international competition at Cheltenham, where they will be up against the best of the world, and Australia has a good track record in this, and I wish them well. They do get also $1,000, and obviously it's for the money, right? This is why we're all doing it, right? Now, they have 3 minutes. That's basically it. There's almost no rules about this. If they want to do poetry, slam poetry, if they want to tap dance, whatever they want to do, it's up to them. They have 3 minutes to communicate that science. Our judges will be marking them on the 3 C's: content, clarity, and charisma. This is somewhat subjective. Maybe you don't agree, but that's okay, because you get to vote too. And indeed, to our viewing audience streaming live on Australia's Science Channel, you all get to vote for the People's Choice Award. I'll tell you a little bit more about that at the end. But just essentially keep your smartphones on, keep them on silent, but keep them on. And keep tweeting about the event. The hashtag is FameLabOz. You can tag AU British on Twitter. Just mention British Council Australia in your Facebook post. We'll pick it up. Basically, keep tweeting, and we have a Twitter wall, and you get to see yourself. You can be part of the action. That's what I'm saying. There's the incentive. Keep your social media active. Now, tonight really wouldn't be possible without our sponsors, and I'd like to go through them all and name them all and thank them all. Our FameLab founding partners, the West Australian Museum and Foundation for the Western Australian Museum, and the McCusker Charitable Foundation. Would you please thank those both? I tell you what, we might actually have to keep the applause going. So, and I'm going to go rapid fire. Our FameLab major partner, Woodside Energy, round of applause. FameLab University partners, Curtin University, Edith Cowan University, Murdoch University, University of Technology Sydney, University of Western Australia. Our state venue partners, keep it going, Queensland Museum, Museums Victoria, Museum of Applied Arts and Sciences, WA Maritime Museum, our British government partner, the British High Commission, keep going. The WA presenting partner, Department of Jobs, Tourism, Science and Innovation, our training and advocacy partner, Inspiring Australia and SciTech, and our media partner, Australia Science Channel. Thank you all. Whew. Okay. So, the last bit of formal housekeeping. Toilets. Back up that way, down the stairs to ground floor. Emergency exit. Back up that way, down the stairs, past the toilets, run outside. Okay, it's pretty simple. Now, we have the hope, the desire that you help us trend online, as I've mentioned. Keep using the hashtag FameLabOz. I certainly will be doing that. We have 10-minute intervals to break up. 2 of these intervals to break up tonight, so you will be able to time those toilet breaks appropriately. So hold on. We have the first of 3 speakers in this first set to tell us a tale of 2 fishes and how they combat climate change. We have Taryn Laubenstein from the James Cook University and the ARC Center of Excellence in Coral Reef Studies. Would you please welcome Taryn to the stage?
Tonight, I'd like to tell you a tale of 2 fishes, Sheila, the damselfish, and Kyle, the kingfish. Sheila and Kyle are ocean royalty. Sheila rules over the coral reefs. She's a bit of a homebody and rarely leaves her coral castle. Kyle, on the other hand, is more of an adrenaline junkie. His kingdom is the open ocean, where he swims hundreds of kilometers to find his next meal. Kyle and Sheila have pretty different habitats and lifestyles, but they do have one thing in common. Their kingdoms are under attack. Climate change, That voiceless, faceless, but very real threat is changing their kingdoms, making the water warmer and more acidic. And in the coming years, it's only expected to get worse. So what is climate change doing to Kyle and Sheila? And can they stop it? That's where I step in. My research looks at how fish from different habitats and lifestyles respond to climate change differently. Because just as two people in the same air-conditioned building can feel perfectly fine, thank you, or in the first stage of hypothermia, so too can fish respond to changes in their environments differently. So to see how Kyle and Sheila are affected by climate change, I raised hundreds of their siblings, tiny juvenile fish, no bigger than your pinky finger, at either current day conditions or under future climate change conditions. And then I measured their performance across a range of behavioral and physical traits. What I found does not bode well for our fishy friends. Kingfish became more active in warmer waters. That's a problem because it can expose them to higher rates of predation. For damselfish, it was their sense of smell that became impaired in acidic water, and they actually became attracted to the scent of predators and injured sibling fish. And for both kingfish and damselfish, they had less energy for growing, developing, and reproducing in warmer and more acidic waters. So it seems we have a king and damselfish in distress on our hands. How can these fish cope with climate change? Well, Kyle's at an advantage. His kingdom, the open ocean, is huge, so he can swim south to avoid warming waters. But Sheila can't leave her coral castle, and neither Kyle nor Sheila can outswim acidic waters because they're everywhere. No, swimming isn't the solution. What these fish need is a knight in shining armor to save them from climate change. And that knight is us. Yes, we humans can save Kyle and Sheila by reducing carbon emissions. Now, I know that's no easy task, but it is a worthwhile one. Large fishes like Kyle are an important food source for millions of people around the world. And small reef fishes like Sheila support the healthy functioning of coral reefs. So grab your sword and shield. It's up to us to defend Kyle, Sheila, and all their fishy friends so they can survive and thrive in the future.
Judges, 2 questions, just 2.
Fantastic, a great response from the audience to that. Tell me, what happens when they grow up? Because some fish actually change sex, don't they, as they mature. Do you know anything about whether these species do normally and whether this could be affected by climate change? So we might end up with too many guys or too many gals in the ocean.
You know, that's a great question. I don't specifically look at sex changes in fishes. I know that's very common in turtles. And actually, there's been a lot of research that turtles are actually becoming much more female because more females hatch at higher temperatures. As for fish, the fish that I studied don't do sex changes, so that doesn't affect them. But that's a really interesting question, a good avenue for future research.
Thank you.
Barramundi do, though, don't they?
They do. That's right.
Hi, Taryn. You spoke to high school students this afternoon and this morning, and obviously climate change is an area of science where they are very engaged. I wondered what kind of questions they asked you and what you told them.
I got really good questions, actually. There was one person who asked whether, because he knew that the climate has changed in the past, and he said, well, fishes were around in the past, so why is it different now? Why shouldn't they be able to survive? And that's actually a pretty common question in climate change research. People say, oh, we know the climate changed in the past. What about ice ages? You know, we know climates change. Should the animals be fine? And what we always go back to is the pace of that change. So in long-term history, sure, we've had ice ages and it's been hotter than it is now, but it was always a slow change. And what's changed now is that the pace of warming is just so much faster than anything we've seen in, very far back history. So I was pretty impressed it was a very young student who asked me that. Yeah.
That's fantastic. Thank you.
Thank you.
All right, thank Taryn. Well done. And let's keep the applause going for our next speaker, Sanne Strongberger from Bond University, who will tell us, can allergy drugs treat bladder disorders? Please welcome her to the stage.
Have you ever had a cough on an allergy that simply wouldn't go away. A big reason for that is the release of histamine in your lungs. Histamine is a chemical produced by your immune system, and you can think of it as your bodyguard. If something is bothering you, it will come along and get rid of it. And whilst the body has the best intentions to keep you safe, it kind of overreacts sometimes and gives you all these symptoms that you normally associate with allergies, like sneezing and coughing. But it can not only affect your lungs, it can also affect your brain, your skin, your gut, and your heart. But what about the bladder? There's a syndrome that I'm particularly interested in called overactive bladder. And it is to do with involuntary contractions as it fills up with urine, presenting as that urgent desire to pee. And that happens frequently, both during the day and nighttime. And it's not just about the physical symptoms. People with overactive bladder are less inclined to leave their homes to go socialize with their friends and do everyday activities for the fear of not finding a toilet when they need one. So how do we help people with overactive bladder if we don't know what causes it? That brings me back to the topic of allergies. If histamine can cause such a strong reaction in your lungs, presenting as that urgent desire to cough, could the same response also happen in your bladder? So I went out to find out what happens if we add a little bit of histamine to your bladder. I prepared pieces of little animal tissue, different doses of histamine, added them to the bladder, and found nothing. There's absolutely no response to histamine, and I tried a few times, a few different ways, and there was still nothing. As I was about to give up on my experiments, I had a thought, how does histamine actually work in the body? Well, when the cells are activated that contain histamine, it is released in a really high concentration in a localized area. So what if I, just before I finish it all, put a single dose of histamine and see what happens? And boom, there it was, a strong response to histamine, something similar that we could observe in patients with overactive bladder. So my research considers the use of medications that have already been tested to be safe in humans and are on the market, more specifically antihistamines. Developing new drugs takes a lot of time, a lot of effort and money, but repurposing existing drugs for different disorders built on previous research is known to be safe in humans and can lead to clinical trials much quicker. So if I can continue to show the association between histamine responses I have observed in my research to what occurs in overactive bladder, we might finally be one step closer to finding alternative treatments for the millions living with this disorder.
Zane, I enjoyed that talk wonderfully. Is the problem age-related at all?
It is more common in older people, more common in females, but the thing is, there's common myths associated. Oh, it's just a normal part of aging. Oh, it's just a normal part of being a female. It's not. And people often don't seek the treatment just because of the perception of it. It's not normal and it's treatable, well, to a certain extent, and definitely associated with aging, but definitely not normal.
I really enjoyed that, and it's putting together two areas in a very, I think, imaginative way. How close are you to be able to do some clinical trials on this so we can really see if it's making a difference?
I'm not sure about the clinical trials yet, but I guess our next step would be to go to doctors that treat patients that have overactive bladder and just ask, have you noticed during hay fever season, do you have less symptoms? So sometimes people just don't realize they're so preoccupied, I have this allergy, and they don't realize maybe their symptoms have actually lessened. So that, I guess, would be our next step, just to see if they have noticed something. Yeah. Thank you.
Okay, and rounding out our first session, we have the ANU's Toby Hendy, who will be talking about poking plants. Please make them feel very welcome.
So if you've had a coffee or 2 or 3 today, then you need to pay attention because your favorite drink is under threat. I'm going to tell you a story. It's an unexpected story. I'm going to talk to you about plants, but I'm a physicist. And I may look nice, but I poke tiny plants with needles all day long. I'm not the villain here, though. The real villain of this story is a disease-causing germ. Now, the plants on this table look pretty carefree, but germs will wreak havoc on their lives. Germs infect plants and cause disease, which you may recognize as spots, wilting, or scabs. Diseases can also ruin food crops, and in fact, up to 40% of crops worldwide are lost to disease. Now, imagine this is enough coffee for everyone in the world. If we were to lose nearly half of this to disease, then a lot of you are going to miss out. Now, you might be able to live without your morning coffee, but this is life and death for the over 1 billion people who suffer from malnutrition. Finding a way to keep plants healthy is important, but I never thought I'd be the one to work on it. I did a degree in physics, studying how forces control the world around us, from the motion of stars through the sky to the path of a ball through the air. It turns out, though, a lot of these equations can also explain things in biology too. Let me show you how. This balloon is a plant cell. Plants become infected by disease when a germ latches onto the side of a cell and then stabs through to wreak havoc. Tragic. Luckily, plants do have a way of defending themselves, and it's called the cell wall. The cell wall is a rigid structure around the outside of a cell. It's like a castle and can detect an attack and send knights to fight off a germ by physically strengthening the wall at that site. In the lab, I mimic germs using tiny needles about 1/10 the width of human hair. I poke plants and see how they respond. I'm interested in the physical factors like the force exerted by the germ, how much pressure is required to trigger the knights to mount a defense, and how long it takes them to fight back. I bully the plants, but it's for their own good. I'm the unlikely hero, creating an army of knights in shining armor who will protect plants, including the coffee bean, from disease. So I hope anyone who has had a cup of it to drink today can appreciate this goal. Thank you.
I don't think anyone needs a coffee after that demonstration. Thank you, Toby.
I'm curious, if you can increase that defensive mechanism and make it stronger, are there negative implications to that?
I guess in terms of biology, everything you do costs energy. So if a plant can have a really strong cell wall, then the energy and I guess resources to build that are coming from somewhere else. So it's going to be a fine balance between defense and other important things you need to do inside the cells.
As a physicist, how were you welcomed by the botanists? Did they think you were a strange interloper, or did you adapt very well, easily?
I am a little bit like a fish out of water, talking to a lot of biologists about this issue because it is inherently a biological problem, plant disease. But I think I've been well received because I'm coming at it from a new perspective, from this physics side. And I think that sort of collaboration is what we need on something that is such a big and important problem. Thank you.
Awesome. Thank you, Toby, and thank all of our first 3 contestants this evening. This is the end of the first session. You can go to the bathrooms. We'll see you in 10. I'd just like to share that backstage energy is building, the excitement's in the air, there's tension. I just saw that magnesium and oxygen had got together back there. OMG. Give me the drums. There we go. Oh, my academic career is down the toilet. Okay, we're moving into the second session. I want to remind you all that you can tweet, as you noticed with the Twitter wall, the hashtag FameLabOz. We also have an opportunity for you, the audience, to ask questions. So as you're watching these fantastic speakers, just write down the question. It comes at the very end, and it's very difficult to remember because you're about to hear a number of other fantastic talks. So write down your questions. And then with, oh God, no, I can't do that joke. That's terrible. All right. I just can't subject you all to that. That was just too brutal. Oh, and of course, you have the Audience Choice Award. Again, especially for those of you at home, you can vote on this as much as the audience here. So again, if there's any you're particularly enjoying, keep that mental balance going and try to keep that weighing up who is your favorite. So, to kick us off with our second session, we have Richard Charlesworth from the University of New England, who's going to talk about how celiac disease diagnosis can be a pain in the posterior. Would you please make him feel very welcome to the stage?
I've brought along a sample of a highly dangerous protein. I'm amazed they actually let me bring it into the building, to be honest. In around 750 million people, that's the number currently affected with celiac disease, myself included, that protein is the enemy of your immune system. When it enters your body, your immune system mounts an all-out attack. It doesn't bar any holds, and it destroys everything that this protein comes into contact with. This essentially destroys the lining of your small intestine. So what is this protein? Well, I've brought along a relatively safe example to show you tonight. This protein is gluten. Now, we know that the celiac reaction is caused by gluten, found in wheat, barley, and rye. And we know that the easiest way to treat the disease is to simply remove gluten from the diet. The problem lies in the diagnosis of the disease. Now, we can diagnose celiac disease by looking at markers in the blood, or we can directly examine the tissue under the microscope. And this is the most conclusive test that we have to date. But there are problems associated with it. It takes a huge amount of skill and experience and a perfect biopsy specimen to conclusively diagnose the condition. Subtle cases can be missed or misinterpreted, and this delays treatment outcomes for patients. So wouldn't it be good then if we had another test for celiac disease, one that didn't have the same shortcomings? This is where my research has been focused. I don't even look at the tissue. Instead, I look at the expression of 87 individual genes all at once in a patient biopsy. I then take this data and I run it through a series of equations that separate patients on a graph. And I've shown that I can diagnose celiac disease in this method with around 95% accuracy, and all without requiring anyone to actually look at the tissue. There's also other things I can do. I can track treatment progression. I can watch patients move on my graphs as they improve. I can potentially separate celiac disease from other intestinal diseases. And most excitingly of all, I can actually detect celiac disease well before it starts to change the shape of the tissue, allowing for much more rapid diagnosis. Now, this work is still in the pilot stages, and it would need to be refined before it could be used as a new diagnostic. But promising results that I've seen so far indicate that this could be the case in the future. So thank you for your time.
Hi, Richard.
Hello.
Can you talk to us a little bit about the difference between real celiac disease, intolerances, lifestyle choices with gluten.
Of
Can you
course.
expand on that a bit?
There's been a lot in recent years about the rise of celiac disease, and it is certainly on the rise. So Finland, for example, around 6% of Finland now has celiac disease. So these are huge amounts of people that are being affected. But we talk about what we call the celiac iceberg. So at the very tip, we have the people affected with celiac disease, that 1% of the population. Under the water, we have this huge kind of unknown mass of people. We know that they're reacting to something in the protein. We don't know what that is. We don't know if it's gluten itself, or whether it's another part of that gluten protein, or whether it's something in the grain, some sort of fermentable product that they have. So overt celiac disease is that autoimmune reaction that we get. All of those other conditions, we're really not sure what causes them just yet.
Richard, you're so confident with what you can achieve in an area that is not only controversial but fashionable. Aren't you a little nervous about making claims in front of all your superior scientists?
That's what we do, isn't it?
Nice. There we go.
Thank you.
Superior scientist, Robin. Up next, we have Pega Masumi from the ARC Center of Excellence for Exoton Studies with her work on solar windows. Please make her feel very welcome.
In a couple of years, we are going to run out of fossil fuels. If we don't cook our planet by that time, we are all not going to die, but we're going to have energy crisis. Dark cities, cold. We probably have limited time of electricity every day. We have to choose either I'm going to work, cook, or to eat, perhaps. So we will be more rely on the renewable energy, solar energy, which is not too bad. So if we can harvest 1 hour of that energy, that would be enough for the entire planet for about a year. But that means we have to have 100,000 kilometers of solar energy being located very close to cities to be efficient. But everyone knows that's not feasible. Our cities doesn't have space. The cities are growing like boxes. And all we don't have is our individual roof. I don't have my own. All the sun I get is through my windows, which my plants can survive without electricity, but not me. So what we're going to do is to make our next generation of solar panels to be more compatible with our infrastructure. What if our windows are working a bit smarter, not just providing the view and sunshine, but also can work and generate electricity for us? Luminescent materials are the materials can absorb the light of sun during the day and then give it back with a slightly different color. We can see them in the glowing worms, in the dark caves and the rainforest when it's dark. If we use these materials with the combination of the transparent material like glass or plastic, and we use them in the windows, they can still be see-through. They can pass the sun through and then absorb it and emit it. And because the light is traveling in the air and inside the slide with a slightly different speed, the light which is actually captured inside is going to be trapped and redirected to the edges. So all the sun is going through this, is going to be redirected to the edges. We call this technology luminescent solar concentrator, or LSCs. So instead of having this much area of solar panel, we will need to have this much area of solar panel. My research is to optimize this technology to be used in our next generation of solar panel and mass production. Maybe we have solar windows. And with this technology, at each corner of the city, we can generate electricity, even the sound barriers in the highways. This will provide electricity to each individual person cheap. Thank you.
What important research. Congratulations.
Dreamy.
Now, how expensive is it at the moment? And can you give us a projection of the likely cost, say, in 10 years' time? Will we all be able to afford this?
Okay.
The planet can't do without it, perhaps, but can we afford it?
That's one of the questions that I avoid to answer. So this is at the beginning of the research. Obviously, if we are going to make a mass production, we need to make it cheap. So these are the coasters that our center made just to advertise our center, and that cost us 10 cents each in China. But that means they don't have the efficiency that we want. So the ones that have the perfect efficiency, they really need to be also cheap, and I don't know when we achieve that, but we definitely need to make it cheap enough that we can use it in the infrastructure.
How can you make that part of the buildings, part of the infrastructure, rather than separate as a place?
So we just need to change the windows. So that won't be the traditional windows that we have. So what we need to do is infusing the luminescence material into glasses that we use in the windows, or putting perhaps a layer of this material on top that can absorb the light and capture the light. So that would be basically based on the manufacturing of the windows, I would say.
Well, our windows are all vertical. Surely you'd need them at an angle to catch the light better. So do you need to get together with architects and completely redesign our houses?
Well, in different hemispheres, we get different sunlight during the different seasons at different directions. So even at the roof, when you have a triangle, the engineers will come and say, okay, put it at the south part and west part, or put it at the north. And so it all depends on which hemisphere you are and how much sun you get and how long that part of your house will get the sun. So yes, that should be engineered as well.
Thank you.
All right, next up we have Murtaza Rezai from Curtin University and the Autism CRC on his talk, Empowering Beautiful Minds. Please make him feel very welcome.
Good evening. Thank you for having me. We live in a world that is highly unpredictable, chaotic, obsessively social, and just turned up too loud. For people with autism spectrum, it is highly exhausting to operate in this world. Autism is defined by limited communication and social skills, repetitive behavior, and high sensitivity to light, noise, touch, and smell. Despite these traits, people with autism have extraordinary capacities to focus, high attention to detail, and passionately curious minds. These traits make them perfect candidates for roles such as data analysis and software testing. Yet tragically, very few successfully transition from primary school to high school. Even lower numbers graduate from high school, and an even lower percentage go to university or have full-time employment. There are many aspects of our world that enables this continuous isolation and wasted potential. But we found that one of the biggest woes is transportation. You can have the perfect job or education program, but if you don't have the transportation to get there, all of it is obsolete. Today, I'm going to discuss how we can make independent travel possible through public transport. You see, public transport is a minefield of anxiety, stress, uncertainty, and sensory overload. For most of us, the erratic bus schedules, overcrowded trains, noisy passengers, and the multitude of sensory stimuli are merely inconveniences. But for people on the spectrum, these inconveniences can cause debilitating anxiety and, more importantly, discourage or limit any attempts at independently engaging in community activities. But our research can change this. My research team and I have worked closely with people on the spectrum to understand their special needs and requirements when using public transport. Some major concerns include, I'm afraid of overcrowded spaces. I have difficulties transferring between services. I'm afraid of missing my stop. What happens if I have an anxiety attack or a sensory overload? How do I communicate if I don't have the capacity to, if I don't have the verbal capacity? On the other hand, for parents, they're worried that their child is going to get lost or get hurt. So we've researched and addressed these concerns into a mobile app that makes public transport more personalized and less stressful. Eliminating or even managing the transportation problem can be a stepping stone to building a world that doesn't allow the wasted potentials of people with autism. A world that will empower them to excel independently at skills that are tailored to the autistic mind. Thank you very much.
Thank you. I love this. I want to know a little bit more about when you tested the app, which specific elements of the app have people on the spectrum found the most useful?
Well, we haven't actually have the full data on the testing. So the testing, we've just begun the testing at the moment. So we will know that very soon, but I'm afraid I don't have a definite answer for that at the moment.
Some of the practical ways, can you tell us then some of the practical ways in which it works? So for example, how does it respond to a change of timetable for somebody?
So one of the biggest problems for people on the spectrum is uncertainty, right? So your bus is running late. For most of us, it is running late. For people on the spectrum, it helps to know that, the bus is coming, it's 10 minutes late or it's 5 minutes late. And so that means I'm gonna be late to work, 10 minutes late to work or 10 minutes late to school. So that is one of the features, providing real-time data. For example, if you're sitting next to someone who has really strong perfume on and that bothers you, so that causes a sensory overload for people on the spectrum. And so we've got evidence-based tips and strategies on how to deal with those situations, or if the train is overcrowded, if there's no seat available, we actually, one of the features of the app is we predict if a service is going to be crowded or not based on historical data. So that will help them make planned journeys that are more personalized, so they can avoid crowded services or they can avoid, yeah.
Thank you. And I love this research. 1 in 68 young people are diagnosed on the spectrum at the moment, so it really is a big problem. But as you say, such talents and such potential. I think it applies to people on the spectrum, but surely this is useful for far more people who want to know when it's not going to be busy. When you're going to take your toddler on the train, when you're going to try and get all your shopping home. Is it something you see that be more widely applicable beyond one community?
Absolutely. So we've designed and built this with people on the spectrum in mind. But we've had conversation with the elderly population to see if they can actually use this app. But at the moment, it is just built for people on the spectrum. But when we release it, when it's out there, anyone can download and use it, and hopefully we can expand on that in the future.
Fantastic. All right. Closing this session, we have Muthu Vigneshi Yale Yapan from Monash University, who's going to talk about groovy patches.
The heart skips a beat when you're excited, bounces when you're in fear, and raises when you're about to propose to your love. Do you know that your heart can pump enough blood to fill an Olympic-sized swimming pool in just 1 year? But when this poor little heart experiences an attack, its pumping efficiency is affected. Love and excitement are replaced by pain and anguish. Heart diseases are killing 1 Australian every 12 minutes. It affects 1 in 6 of us here. Heart transplantation remains the only definitive treatment, but heart donors are limited and most patients die while waiting for a suitable heart. So what are the other treatments then? Scientists have tried 2 different approaches to fix the heart. In one approach, heart muscle cells were injected into the damaged region, but they don't stay put and tended to migrate somewhere else in the body. In the second approach, heart patches were used like Band-Aid, but heart muscle cells don't efficiently align on them, leading to poor healing. Alignment of heart muscle cells is like giving a handshake. If one hand is like this and the other is like this, of course we can give a handshake, right? The same thing happens to heart muscle cells, where they need to be aligned to produce a heartbeat. Hence, my research focuses on overcoming the shortcomings of the current treatment for bringing back the joy to heart patients' life. How am I doing it? I use 3D printing to print with 2 materials. Then I removed 1 material, resulting in grooves. The width of the grooves is 1/10 the thickness of a human head. So how does this groovy patch improve alignment then? Good question. Heart muscle cells are elongated in shape. When they see the grooves, they tend to elongate and snuggle along them. This will result in excellent alignment and then beat in time together. Thus, the groovy patch with aligned heart muscle cells will be used to seal the damaged heart region. This will heal the heart. My hope and my goal is to find a permanent cure for millions of heart patients out there, to let them feel the love of their close ones. Thank you.
Are you feeling the love? Are you feeling the love right now?
Yeah.
You feel amazing.
All right. Proud.
Robin.
Thank you. Thank you.
Muda, congratulations. It's very encouraging. I've had a pacemaker since 1988, and it hasn't worked for 17 years. Can you fix me up after the session tonight? Would it take long to apply?
Definitely, but just maybe 3 or 4 years. If you can wait, because like we are in the process of doing the medical testings. So once it is ready, definitely I'll ring you.
Hope I'll last.
Definitely.
Very exciting research. Thank you for sharing it with us. This applies to heart muscle, but could you apply it to the skeletal muscle? Is it a similar principle for after, say, there's been damage through trauma to say, your biceps or triceps? Or is it only for the heart?
It's a really good question, but my interest relies on heart. So as of now, we are like really working on fixing the heart. The main reason is the shape of the heart cells. It also plays a very, very important role for fixing this. Since it's an elongated shape, it can really work with the groovy patch. But regarding the other muscle cells, we are really not really sure because we haven't done any testing with it. But definitely we'll consider that. It's a very good suggestion. Thank you very much. Yeah, principal's there. Thank you.
All right.
Thank you.
Thanks, Nathan. I don't think I've ever witnessed an attempted bribery of a judge by fixing their heart. I mean, that's brazen. That closes the second session. We have one final session remaining, but for now you have 10 minutes to, well, yeah, go to the toilet, and tweet #FameLabOz. See you in 10.